Okay, so the next genetic engineering technique that I'll talk about is polymerase chain reaction. Now, this is a rad technique that is used to amplify a section of DNA, like a target sequence of DNA, into millions or billions of copies in a very short amount of time. Now, polymerase chain reaction, or PCR, is going to rely on thermal cycling. As we go through this, there's going to be 3 different steps that we're going to focus on. And each of those steps occur at a particular temperature. All right. So if the purpose of polymerase chain reaction is to amplify and make copies of DNA, we need to think to ourselves, okay, well, what is required to make more copies of DNA, to replicate DNA? So in your mind, hopefully you're thinking, well, we're going to need nucleotides because nucleotides are the monomer or building blocks of DNA. Now, you can't just randomly throw nucleotides together to get a specific sequence. You're going to require an enzyme. So the enzyme that adds or synthesizes a complementary sequence of DNA is called DNA polymerase. Now, in PCR, though, there is a very specific polymerase that's going to be used. It's called Taq polymerase. And that Taq polymerase was actually discovered in the geothermal, hot, boiling waters of Yellowstone National Park in 1960s, I think. And why this particular polymerase is important is because it actually doesn't denature in hot temperatures. So if you recall our unit on enzymes, we talk about denaturing. It's an enzyme, and it doesn't denature in hot temperatures, so we use Taq polymerase for PCR. And then if you're thinking to yourself, okay, in DNA replication, there was actually numerous enzymes involved. We are actually only going to use polymerase, Taq polymerase, but we do need to add primers. In regular DNA replication, you would have primase, the enzyme synthesized primers. You would have DNA polymerase 1, remove those primers. In PCR, the scientists are going to take pre-made primers that are the right sequence, that are complementary to the ends of the target sequence of DNA that is intending to be copied. And so they will add primers to the solution. And so here, to begin PCR, we need the DNA that's going to be copied. We need nucleotides, billions of nucleotides, as well as copies of primers and Taq polymerase. All right. Let's go ahead and see the 3 steps involved in the polymerase chain reaction. So first is denature. We're going to heat the DNA sample to anywhere from 94 to 96 degrees Celsius, and that's going to denature the DNA. So DNA is a double helix, so by denaturing it, we're basically breaking the hydrogen bonds that were holding the base pairs together. A pairs with T, C pairs with G. Those base pairs are held together by hydrogen bonds. So this high heat separates the double helix. Then the next step is step 2, which is called annealing. Now in annealing, what's going to happen is we're going to cool the temperature down, I believe, to something around 68 degrees Celsius. And that is going to allow the primers to now base pair with that open double helix. So we separated the DNA, we cooled it down. Now base pairs will form between the DNA strand and the primers. Now, once we have the primers, though, we now have a starting point to synthesize the new complementary sequence to build that double-stranded DNA, and that is the job of Taq polymerase. So, Taq polymerase's optimum temperature, though, is 72 degrees Celsius. So we're going to warm it back up a few degrees so Taq polymerase can work at its full potential, and then it will elongate and make copy or base pair and add new nucleotides to build the second strand of the double helix. Now that is one cycle of PCR, and this actually repeats anywhere to 20 to 30 times, 40 times, 50 times, and basically it leads to exponential growth in the amount of the DNA sequence that you're interested in. So I'm going to go ahead and elaborate and explain this again with my own animated visuals. So here we start with a target sequence of DNA. Now that first step is to heat and denature. So we're going to increase the temperature to about maybe 95 degrees Celsius. That's going to cause the double helix to denature. Now, and separate. And so here I've identified the 5' and 3' ends because now as the primers come and attach in step 2, the annealing step, the primers are going to attach at the 5' ends, right? Because DNA is built from 5 to 3. So here we add our primers, or the primers are added at this cooler temperature, 68 degrees. And then we're going to use Taq polymerase. We're going to, step 3, we're going to heat it back up to around 72 degrees Celsius. And now Taq polymerase can elongate and build that complementary sequence of DNA, build the double strand. So now we have 2 copies of DNA. Awesome. Now that was only one round. So if we repeat this for round 2, here we're going to do step 1, denature. Heat it up, break those bonds. Then step 2, we're going to anneal. We're going to cool it down to 68 degrees so the primers can base pair. Then step 3, we're going to elongate. We'll warm it back up to 72 degrees Celsius where now Taq polymerase can extend and build that complementary sequence. We're at 4 strands of DNA, making copies of that target gene of interest. Now in step 3, we would replicate those 4 strands. So now we're up to 8 copies. And then in the 4th cycle of PCR, we would take those, 8, and then they're going to replicate. So now we're at 16, right? So you can see how over time, if we started with 1 double helix, 1 section of DNA, in the 1st cycle we ended with 2, and then 2 became 4, and then 4 became 8, and then 8 became 16. Now, if you continue this by cycle like 20, you'll have over 1 million copies of your target gene of interest. By 30 cycles, you'll be over 1 billion copies of this DNA sequence. So why is this important? Well, PCR, polymerase chain reaction, is used to amplify DNA fragments into millions of copies. Once scientists have millions of copies of this DNA, they can use it in genetic testing, in research. Ooh, what if it was like an ancient DNA sample? What if it was like, Jurassic Park where there's DNA preserved in amber and they only have a small section? Well, now they can amplify it and they can use it and study it however they want because they have 1 billion copies, right? And so it's also used in infectious agents. Right now, this is 2023, and so we're we've had a lot of experience with COVID and PCR tests. And so when COVID is actually an RNA virus, so there's a couple extra steps involved to do PCR for COVID, but basically they're amplifying any DNA samples taken from your nose or wherever and seeing is that DNA sequence or RNA sequence from COVID present, right? And so an RNA virus is going to have to be a different video of how that works, how you could start with RNA and then work backwards to synthesize DNA. That is a whole nother subject. But anyway, so this is PCR, and it's used to amplify a section of DNA or a gene in a really short amount of time. All right, great job.